listeners, welcome to episode 111 of Cell and Gene, the podcast. I'm your host, Erin Harris, and I have another great episode lined up for you because I had the opportunity to talk to Ernexa Therapeutics president and CEO, Sanjeev Luther, about the company's core technology that focuses on engineering induced pluripotent stem cells, or iPSCs, and transforming them into induced mesenchymal stem cells, or iMSCs, for advanced cancer and autoimmune disease. Sanjeev and I get into how Ernexa's synthetic, standardized, off-the-shelf iMSCs are designed to overcome the historic hurdles in MSC therapy, and that includes variability, scalability, and manufacturing complexity. And they do that by leveraging master cell banks, precision gene editing, and novel product engineering. He also shares his insights into the process of developing pro and anti-inflammatory iMSCs for oncology and autoimmunity. And importantly, I asked Sanjeev for his take on how he sees manufacturing innovations and partnerships playing a role in accelerating the adoption of next-gen iMSC therapy. So definitely stay tuned for that. All right, I will let you get to it. Here is my conversation with Ernexa Therapeutics' Sanjeev Luther. Enjoy. Sanjeev, hi. Welcome to Cell and Gene, the podcast. Thanks for your time.
Thanks, Erin. Thanks for having me.
Absolutely. So tell us a little bit about Ernexa Therapeutics.
Well, Ernexa Therapeutics is developing innovative cell therapies for the treatment of advanced cancers and autoimmune diseases. It's a publicly traded company, and its core technology is based on induced pluripotent, which is iPSCs. And then those iPSCs are a type of stem cells that can be reprogrammed from adult cells to become nearly any cell type in the body. And then that's transformed into iMSCs, which is mesenchymal stem cells, as you know that. So you start with iPSCs, you go to iMSCs, and then they're edited to generate what you call cytokines or interleukins. And there are two sets of these products. One is for oncology, of course, which is interleukin 7 and 15, which are pro-inflammatory products. And then IL-10, which is for the autoimmune side of the business, which is the anti-inflammatory product. So think of it, it's coming off the same technology, but one side is pro-inflammatory and the other side is anti-inflammatory. And I also believe, to the best of my knowledge, I would say to our knowledge, Ernexa stands apart from, I think it's the only cell therapy iMSC company in the US, off-the-shelf company, which is focused on oncology right now, which is iMSCs and this interleukin 7 and 15. I think I would stop there, right?
Yeah, sure. Great. Excellent. So thanks for that thoughtful explanation of Ernexa, and also for the spelling out the acronym of MSC. I was going to do that. So that was perfect. I'm glad we got that out of the way. All right. So talk to us a little bit then about traditional MSC therapy. So they've faced hurdles such as variability, limited persistence, manufacturing complexity. But from your perspective, what have been the most significant challenges with mesenchymal stem cells, or MSCs, to date?
Right. So the MSCs to date, as you know, that they are more bone marrow MSCs. And as I keep saying, we are allogenic, off the shelf. So as you pointed out, there are really four things which inhibit, I think, the development of MSCs at this point. One is variability, persistence, manufacturing, of course, and then the regulatory standards, because it's still an area which is, I would say, evolving, as you know, that from regulatory standards. So it's not very clear exactly where to go. So variability is a very, very big issue right now. Depending on where the MSCs come from and how they are processed, results can be very different. And also the other issue with MSCs so far, not ours, and we'll get into it, single donor versus multiple donors. So heterogeneity versus homogeneous, right? And that's a big issue. And I believe we have overcome that by single clone. The other thing is persistency. At the bone marrow MSCs don't last long once given to patients, sometimes maybe just 2 weeks. So think about it, you may have to do it multiple times. And then manufacturing, I believe, is a major, major bottleneck right now because of that heterogeneity issue, actually. So, and then of course I talked about the regulatory standards. On top of that, the question is genetic modifications of these bone marrow MSCs is very challenging, actually. Which is what Ernexa does, right? So in terms of increasing efficiency of iMSCs, it's very harder to transduce or edit compared to T cells. Getting stable expression out of them without losing, viability and is tricky, actually. And then cell identity is another big issue. And then safety, of course, there are some safety concerns around insertional issues and off-target effects, as you know, that you may have heard about that. And then, of course, the bigger issue is scalability. Can you actually scale them, right? But because of these obstacles, MSCs, while very powerful, as I would say, that have not delivered the therapeutic benefit they are capable of, I believe, you know.
Good. Okay. So thanks for that background, kind of the big, broader background of MSCs, because I would like to now talk specifically about Ernexa and iMSCs. So you're advancing standardized synthetic allogeneic iMSCs. Now, how do they address some of the core issues we've seen with donor-derived MSC therapies, perhaps such as batch-to-batch variability, scalability, the things we've been discussing?
Yeah. So I'll touch upon those 3 points, which I touched on the other side: consistency, scalability, and reproducibility. You know, that's really the way where our focus is right now. So iMSCs come from a defined, so our Ernexa's iMSC come from a defined iPSC line, actually. So it's a single defined iPSC line. So you don't have the donor to donor difference. It's single donor, single clone, right? So it's not like you're taking this donor, that donor. That makes traditional MSCs really unpredictable, but these are very predictable because of that. Scalability, you know, so once you establish the gene, right, in our case, edited iPSC master cell bank. So, you know, so the way our thought process works is our iPSC is master cell bank 1, our iMSC is master cell bank 2, and then IL-7 and 15, what you give to the patient is master cell bank 3. So it's a very simple process. And so that's why there is no variability from batches to batches because of that. And then, of course, the reproducibility is a very important thing. So every batch can be made under same control process, actually, which helps meet regulatory standards much easier. So that those 3 things sort of set us apart. And then, of course, the engineering potential, so our iPSCs can be genetically modified as early so that edits carry through consistency into iMSC. So think about it, that you're actually editing the iPSC cell line, not the iMSCs, actually. So that editability continues on from master cell bank 1 to master cell bank 2, and then to master cell bank 3, right? And then there's supply chain advantage, no reliance on multiple human donors, you know, and which reduces variability and logistical hurdles, single donor, single clone, right? So that's, I believe, you know, time will tell, of course, but based on the preclinical data to date, it shows that those are the advantages of our iMSCs versus the other currently available iMSCs. Yeah.
Sure. Okay. I want to talk a little bit about how synthetic and engineered, the nature of the iMSCs enhance their therapeutic potential compared to conventional MSCs in oncology and autoimmune disorders. So oncology and autoimmune, very broad, but you know, can you talk us through that a little bit?
Yes, this is a challenging topic, actually, to get into it, but I'll do my best, right? So, you know, I always talk about what is the secret sauce here, right? The secret sauce is the modular plug and play nature of the iMSC platform. So it's a plug and play platform, right? That we so far have talked about, which enables us to leverage the intrinsic homing ability, which is very important, and the immune self engineer ability to selectively deliver the potent pro or anti-inflammatory. Remember, I talked about pro-inflammatory or anti-inflammatory, right? So it allows us to do that and then target the tissues, limiting systemic exposure. Very, very important. Also, the ability to easily engineer in combination of factors. So, you know, so for example, IL-7 and 15, IL-4 and 10, IL-12, you can actually combine these things, you know, very easily depending on which disease you are going after. So, for example, on oncology side, we took 7 and 15. They are not given separately, actually. They are actually linked. It's a fuse 7 and 15, and you're able to do that versus if you look into where the market is, there's a lot of IL-15 being used, IL-7, but separately. So I think we may be the first one who has brought this 7 and 15 as a fused product together, you know, and we see definitely distinct advantages in terms of efficacy of this fused product versus 7 and 15 separately given you. And then another I think the thing one should try to explain is the foundation in iMSC. What is our biology behind it, right? At the end of the day. So conventional allogeneic MSCs already show promise across many diseases. Like conventional MSCs are showing that. So their impact comes both from modulating the immune system and from their unique ability to home. to sites of inflammation in case of autoimmune diseases and avoid rapid immune clearance, actually. So that's one of the biology we are very much focused on. So what makes iMSCs different, right? With iMSC technology, we take those natural strengths and engineer them from consistency and precision. So very important is that consistency, as I've been talking about. That's a pretty big challenge, and we quite heavily focus on that issue. Rather than being a one-size-fits-all, iMSCs act as an adaptable and programmable delivery. So it depends on which disease you're going after, right? So, for example, if you go after Crohn's disease, maybe we have to modify our IL-10 product to go after that, right? And then, in fact, we're looking at that. It won't be just a base IL-10 product. It has some modification, which we are making it to from an editing point of view. So why this matters therapeutically is very important. iMSCs can deliver therapies directly to diseased inflammation tissue, right? So, for example, in ovarian, we are doing intraperitoneal, right? So it's right in the peritoneum. That's where you're delivering this IL-7 and 15, right? Where they're most needed in this case, right? Same way when you go to the autoimmune side, it's at the joints, right? So how can you deliver right there, right? So the question is, can you unlock new possibilities from this kind of technology? And this opens the door to using biological agents that are powerful or toxic. So the other thing is you can combine these with other biological agents also very easily, right? Because since you can do 715, now you can combine it with another product, right? So that sort of gives us what I call a little bit of I would say it gives us a little bit leeway to go left or right as which disease you're going after. So in both in oncology and autoimmune disease, that precision and adaptability could be game changing actually. Now, of course, time will tell. As I said, so far in the preclinical studies for both sides, both disease areas, we feel very, very encouraged right now, what we are seeing.
Good. Okay. I want to talk, you mentioned certainly scalability at the top of our discussion, and I want to circle back to that a little bit. Scalability, patient access remain challenges across the cell and gene therapy space. How would you say that off-the-shelf iMSC therapies can help shift that sort of paradigm from highly individualized treatments to more accessible options for much larger patient populations?
Yeah. So that's an important question. So let me just give me a second. Let me get there. So the one thing maybe I should clarify further. So some of the challenges which you're talking about, I call it the N of one, right? So it's per patient you're trying to do it, and that's where the cost is much, much higher for CAR Ts, for example, right? One thing to keep in mind is we are allogeneic and autologous, right? So that's a huge difference, right? That gives you a lot more leeway. And as I said, allogeneic cell therapies have the potential to overcome some of these challenges which you just brought up. They provide readily available off-the-shelf product. Remember, I said that we are off-the-shelf synthetic, right? So that is a huge advantage. You don't have to keep looking for a specific, thing. It's off the shelf, and it reduces manufacturing complexity. We are actually in the middle of doing we have started that process now, and we find that it's actually a lot easier compared to other products which the same manufacturer is manufacturing, right? And we hear that constantly. And also it can enhance the treatment accessibility by reducing turnaround time. The turnaround time. So remember what happens. This is not like you have to draw the blood of a patient and then you do the edit and then you send it back. No, since they're off the shelf, they actually come in a vial. Right now, for our preclinical work, they come anywhere from 1 million to 5 million cells in a vial, right? Mm-hmm. So the idea would be the same thought process, right? As you go to the patient, right? That you will be able to send them like a drug is sent, right? So from the manufacturer facility directly to the patient center, right? You're able to deliver these cells. It's not like the turnaround time between drawing the blood, bringing it back to edit, sending it back to that. None of that is needed actually. In fact, once the product is ready, it's ready to be given to the patient, right? And the duration of that, the whole time to express it from that iPSC to iMSCs is only 44 days actually. Within 44 days, you're able to do it actually. So the gene editing, like knocking out of deploying immune evasive markers. So some of those things you can do with this technology. And then iPSCs, as I offered, is a very scalable, uniform source for off-the-shelf therapies, right? And so that's, I believe, is the single biggest attribute of our technology, which sets us apart from the existing iMSCs and existing autologous products and so on and so forth. And that's partly the reason I'm sort of trying to, or the company's trying to move in that direction. The other thing is that this also allows us that shift from N of 1 to 1 patient, right? As I keep talking about, today's manufacturing models are largely built around autologous and equal ones therapies, right? And to reach the scale needed for broad patient access, the entire manufacturing mindset must evolve. Because if you continue with that mindset, the scalability, the reach, and access and cost will continue to be a huge barrier, right? So innovation in automation and bioreactor design and quality control are critical for iMSCs, and that's one of the reasons we chose the manufacturing facility which we have chosen. I mean, I'm almost there. That's why I'm not going to take that time. Almost there. Who's going to manufacture this product? And so I would say those are the things which overcome the current challenges of the available cell therapies, and especially gene therapies. And one of the reasons that's why I'm focusing very much on the cell side of the business right now.
Good. Okay. Now of course, you mentioned manufacturing innovation, which I do want to dig a little bit deeper into before we wrap up our discussion. So, and yes, I agree with you. The mindset certainly has to evolve and change. So what does that actually look like, though? That's kind of what my question is here for you. What role do you see manufacturing innovations and partnerships playing in accelerating in the adoption of these next-gen iMSC therapies? But what does that really look like?
I think what it really looks like, it can't be just a general CDMO. When you're in small molecules, all you do is go to a CDMO. Somebody manufactures your API, and then somebody manufactures your drug product, right? So drug substance. By the way, that concept exists in cell therapy too. There is a starting material, there is an end material. So there is a drug product, like in this case, IL-7 and 15 will be our drug product, right? You could argue your iPSC could be your drug substance. I mean, you could argue that, right? So what needs to happen? It has to be. I give you an example. We are going through a tech transfer right now. It's no longer what we used to do on the small molecule side of the business. You take a SOP and you send it over. It doesn't work that way in this business. It's not an SOP. Actually, a physical person from the company who does all this work in the company today, the editing of the iPSCs, the editing of that individual has to be physically at the CDMO actually. Mm-hmm. And so what I'm trying to get at is that the way you're going to overcome this is it's not a traditional CDMO. These are specialized CDMOs who focus on cell and gene therapies, who actually work on partnership basis. It's not about let me just flip over the stuff to you. It's right. It's a lot of back and forth, and in fact, that part probably takes longer than to actually do the tech transfer, because we are in the midst of that right now, right? Mm-hmm. So I think to answer your question, it's going to be the kind of facility. So for example, you want to go into a facility which has multiple clean rooms. Can't be just one clean room, right? They have to have, because the same cell therapy organization is doing CAR T, sometimes they're doing NK cells. And now suddenly you come up with these iMSCs, which are allogenic. And the one thing is interesting, given that I pointed out that we are the first one with this kind of technology. So we are the first one sometimes at the CDMO too. As I was looking around for the CDMOs, a lot of them had never worked on it, of this kind of a technology. They have worked on iPSCs, but have they gone all the way to this editing iMSCs and this master cell bank 3, which is IL-7 and 15? The answer is no. So I think we are the trailblazers there. In fact, we may be the first one to doing this at the CDMO, actually.
That's really interesting. And so just out of curiosity, because you are in fact the first, what do those early discussions then look like from your perspective to the potential CDMO partners?
I think it's a two-pronged approach. One, the regulatory side has to be also involved. It becomes very important, right? Since you're the first one, right? I would say usually somebody like me who's not a manufacturing guy, I wouldn't be visiting facilities, right? I've been to this facility now 3 times myself, right? With our CMC head, of course, right? And the thing is, and you have to look at every nook and corner. Where do they do their QA? How are they going to transfer the product? How the product will be intaken, right? How it will be processed? How is the process development? How is the scalability going to be done? I had never gotten involved so much in manufacturing before. This is probably the first time I've gotten my hands really dirty. Actually, I understand it now, what this is. But that kind of partnership is needed. So there's the CDMO CEO, myself, we are actually in the room together several times now, going through this
Excellent.
thing,
Yeah, sure. Makes a lot of sense. All right, listeners, we are at the end of our time with Ernexa Sanjeev Luther. Sanjeev, thank you so much for your time and all the expertise you shared with us today. This has been a really thoughtful conversation. I appreciate you.
Thank you, Erin. I appreciate it.
Thanks for tuning in, listeners, and be sure to visit us at cellandgene.com and subscribe to Cell and Gene, the podcast, wherever you get your podcasts to automatically receive every informative episode just like this one. And we'll talk to you soon.